Welcome back to your class in microbiology. For this meeting, we will discuss microbial control or how to prevent the growth of microorganisms. So we have two topics under this. You have the physical method of microbial control and also likewise the chemical method. Let us try to look first into the history how microbial control was practiced. So surgeons in the mid 1800 or year 1800 opened the pens wearing their street clothes without washing their hands. That is the life of a doctor then. They just simply wear their daily clothes and do not frequently wash themselves, not even their hands. They frequently use ordinary sewing threads to suture wounds and stuck the needles in the lapels of the frock, this portion, in their frock coats in between patients. So if a surgeon will treat or work on operation to patient X without wearing any laboratory gown, without using alcohol lamp, without using alcohol even, or any precautionary measure to prevent infection, was not a practice during those times. And the sterilization of what they're going to use, like the needles, was not even practiced. Surgical dressings were often made up of surplus cotton from the floors of cotton mills. So it was not also sterilized. It is just an ordinary thing that is considered as a piece of cloth. It was against this background that French scientist Louis Pasteur, if you can still remember him, demonstrated that invisible microbes cause diseases. These are unfamiliar things that could be seen as an ordinary thing during the early period of medicine. Vast majority of emergency patients in 1900 made their way to the hospital in horse-drawn ambulances. The motorized ambulance made its debut in 1899 when a Chicago hospital adopted an electric version, and the breakthrough found its way to New York City the following year. But this is how surgery were made. It is open to public. This is actually the surgeon, and this is the audience. So for medicine doctors or even interested persons about a certain medical activity on surgery, they are seated in this manner and they are exposed directly to the patient and the doctor working with the surgery. So in a close-up, you can observe that the surgeons or the physicians just simply work on the table with the patient without wearing any mask, without wearing any laboratory gown. The surgical team wasn't alone in tracking contaminants into the operating room. Unlike the scrupulous, sterilized modern operating room in the 19th and 20th century, many procedures took place in large, open-air operating theaters filled with no barriers between the patients and spectators and clothes. Since early electric lights did not always give off enough light for surgery, these operating theaters included large windows to let extra sunlight. So you could just imagine that. In the 19th century, surgery was risky and dangerous. and patients undergoing even the most routine operations were at very risk at high temperature. It was so because surgery was not performed under sanitary or aseptic condition. The operating room, the surgeon hands, the surgical instrument were laden with microbes, which caused high level of infection and mortality rate. Surgeons in the mid 1800s, they often operated wearing their street clothes or daily clothes without washing their hands. They frequently used ordinary sewing thread to suture wounds and stuck the needles in the lapels of their coats. Surgical dressings were often made up of surplus cottons or jute from the floors of the cotton mills. It was This is the reason then that Louis Pasteur and other scientists had worked on the possible occurrence of microbes that could further worsen the disease. So this is an example still of a photo showing to you how surgery is done. So you can see here the physician holding a knife, and you have there the cotton, an ordinary piece of cloth held by one person, and another doctor showing that directly without any gloves or their bare hands is used in the touching or treating the wound area of a patient. Now let's go to reality. Microbiocidals. Microbiocidals from the word or from the suffix "cidal", meaning to kill. So microbiocidal cause microbial death. Another terminology that you should also know is stasis agents. Stasis. They slow or retard growth but do not kill microorganisms. We have also microbial death. Microbial death refers to the permanent loss of reproduction capability under optimal growth condition, or that is the zero increase in number of microorganisms. That is microbial death. As compared with stasis, there are still some microorganisms present, but the growth was only slow. Now, what is sterilization? It is the complete destruction of all microbial life. All or nothing, something can almost be sterile, just like you can't be almost pregnant. So if we say sterilization, it is all or nothing process. All microorganisms were killed. Nothing was left. So there is no such thing as almost sterile. Sterile is sterile. No, not somewhat, not maybe, a little bit. That's not the case. It is absolute. So it is either life or death, pregnant or not pregnant. Because there is no such thing as she is slightly pregnant. There is no such thing as she is slightly dead. Once dead, it is dead. Once pregnant, it is pregnant. This is the same in sterilization. Since time immemorial, heat is almost the most common method for sterilization. Now let us try to uh, make some uh, things to differentiate what is disinfection and what is antisepsis. Because sometimes a disinfection is referred to antisepsis. And antisepsis, just as the same, was also called disinfection. But what is the difference? They are both removal of harmful organisms from the surface, any kind of surface if you are referring to disinfection. You can disinfect the laptop that you are using now. But if we say antisepsis, you are referring to the removal of microorganism from one's skin. So antisepsis refers more to human. Disinfection refers not only to human but also to other things like the table, the laboratory table that you are using. Every time you conduct your laboratory experiment in microbiology, you have to disinfect the laboratory table. But we do not say you antisepsis the laboratory table, but we say disinfect the laboratory table. Disinfect the glasswares that you are using at home. Disinfect the spoon and fork that are used in the restaurants. However, both disinfection and antisepsis go for not sterile. Their product is both not sterile. Generally involves the use of chemicals, that is disinfection. But since we are referring to living organism in antisepsis, they use less harsh chemical substances like rubbing alcohol, hydrogen peroxide, or your what you call this, the hand sanitizer. These are the things that are commonly used for antisepsis. And you are familiar with antisepsis because you are living in the times of COVID quarantine period whereby you are required to practice antisepsis. That is, living with alcohol in every establishment that you go. As you enter, as you leave, you are required to apply alcohol to your hands. But as a tip to you microbiology student, if you are using the alcohol, do not only limit the application of alcohol in your palm area until this portion only. If you could do until the near the elbow, then do it. And then for disinfection, uh, example is the best pin. This is the most common used thing in hospitals and clinics, and even in most laboratory rooms or establishments, because there are some laboratories that are specialized with microorganism or medical analysis. So for antisepsis, any antibacterial lotion or liquid, like as I mentioned, the alcohol. Now, what do we mean by the suffix "cidal" and "static"? Bactericidal. Cidal means to kill. Bactericidal are agents or chemical agents that kills bacteria. Static means to prevent or to inhibit. Bacteriostatic. Bacteriostatic agent will only inhibit or prevent the bacterial growth. Growth will resume after the agent is removed, either bactericidal or bacteriostatic, because its effect is no longer that effective. There are different methods to destroy or reduce. the growth of microorganism in a certain area. It could be physical, it could be chemical, or it could also be mechanical. Primary targets are microorganisms capable of causing infection or spolates are the following: vegetative bacterial cells or the endospores, fungal hyphae and spores for the case of yeast. So they are more of the appendages. And then protozoan, trophozoites, and cysts, worms, viruses. These are the primary targets of microbial control. Cellular targets of microorganisms includes not only the appendages, but also the cell wall, the cell membrane, the nucleic acid, and protein. Here is an illustration telling to you the option for microbial control. First, microbial agents stop attachment. So if attachment to the skin was prevented, it stops growth. Or directly, the microbial agent stops growth. Or sometimes it stops the contact. However, a better microbial agent should kill, not only stop, not only prevent the entry, but it kills already. Okay, this is the hierarchy of resistance. At the bottom, you have there the least resistant. As you go higher, you have moderately resistant. And then the highest of the hierarchy are the most resistant. So, what are the least resistant targets? The vegetative bacterial cell. And then moderately resistant includes the cyst, the fungal zygospores or the spores itself, or even the naked virus. That is the most favorite thing for a virologist to look into because that is the easiest way to kill virus or stops the growth or DNA multiplication of virus. And the most resistant are the bacterial endospores. So the types of control that we are going to focus on includes sterilization. Now, what is sterilization? Is sterile. It is the process to destroy all what I mean here is all viable microorganism, including the virus and then the spores. And what is used here are the microbiocidal. Disinfection, a process to destroy vegetative pathogens, but not endospores. Disinfection is used or applied to inanimate objects, like the ball pen that you are using. If you apply alcohol, that is disinfection. To an open wound, if you apply hydrogen peroxide or the agua oxigenada, that is disinfection. Antisepsis, hence, we are trying to disinfect a certain exposed body part. Sanitation, this is the process of cleansing technique that mechanically removes microorganisms. So washing, just simply washing is a form of sanitation. Covering your mouth is a sanitation. Using gas mask, using face mask, using face shield is a form of sanitation. Then have the term or accept the term degermanization. Degermanization is the reduce in the number of microorganisms. Okay, there are different factors that influence the effective effectiveness of microbial control agent. The number of the agents, or what I mean, the number of microbes. Of course, the greater the number of microorganism, the more that you need a stronger microbial agent. Nature of microbes in the population. So if you are trying to kill something that survives in low temperature, of course, you do the reverse. You increase the higher temperature to lose that cold temperature. So cyclopiles could be killed with just simply increasing the temperature. Just as the same with thermophiles, how to kill the thermophiles, you subject them to low temperature. So another is the temperature and pH. I'm discussing that already. Concentration or dosage of the agent is another thing that influences the effectiveness of microbial agent. But it doesn't necessarily mean that the more alcohol that you place into your hand, the more microorganisms are killed. But rather, the concentration means using 40% alcohol, 70% alcohol, and 95% alcohol. Of course, um, we are using the 70% alcohol. There is still a higher concentration, but that we do not already encourage because it could physically destroy already the skin. Okay, what are the physical control practices that we are using? Or we are referring to the physical methods to control microbial growth. First is heat. If we say heat, it could either decrease the moist or make that inanimate object dry. You have learned very much that in microbial nutrition, water is needed by most microorganisms to survive. So if you're going to dry or you're going to increase the temperature, microorganism could be killed. Our simple practice of exposing your pillows or the the things that are present in your bedroom after a long uh, rainy season is already a physical method of microbial control. So after this class, if you bring out the pillows that you are using, you are practicing microbial control. Another is filtration. This is commonly used in the treatment of the drinking water that are sold in the market. One of their process of killing microorganisms is filtration. Although there are many uh, practices that they are using in order to make the water that you are drinking safe from microorganism. Just one only is filtration. And then low temperature. The food that we are uh, buying in the market are usually placed inside the temperature. That is to prevent spoilage. But putting that means lowering the growth of microorganism or even killing some microorganism. Another is desiccation. Desiccation is the removal of moisture. And then osmotic pressure, that is exposing the object into high pressure contributed by salts. And then radiation. This is commonly used in hospitals. They make use of light radiation or UV radiation in order to sterilize rooms of the hospitals. And then we have also heat-based microbial control process. The first picture on the screen is actually an incinerator. In the laboratory, we make use of the alcohol lamp because we are not yet that rich in order to purchase incinerator. But inoculating loops and inoculating needles, uh, in clinic laboratories make use of that kind of incinerator. Autoclaving. Of course, you have an autoclave in the laboratory. And luckily, your subject biochem should have already introduced you the autoclave that we are using in the laboratory. We have that. UNP have that. And then another is pasteurization, as introduced by Sir Louis Pasteur. So both autoclaving and pasteurization uses high pressure, high temperature, but low time exposure, or just simply short time. Boiling disinfection. However, I'd rather go for autoclaving or pasteurization if we are doing laboratory works rather than just simply boiling. This is the autoclave. This is the kind of autoclave that we are using in the lab. What kills the microorganism is the steam under pressure. The temperature is above 100 degrees centigrade. That's why we are able to produce steam. Very high penetrating power because of the pressure given. Those are the things that kills bacteria and spores, leading to the denaturation of. You need high temperature, you need high pressure in order to denature the protein. Typical cycle for autoclave or autoclaving is 30 minutes at 1 atm or 18 psi at 122 degrees centigrade. That is the capacity of our autoclave in UNP lab. The items to be sterilized must be heat stable. That is the very reason why it is Sir Adonis that usually works with you if we are working with autoclaving, because he checks whether the items that you are going to place inside the autoclave is properly packed or properly labeled also. And the position or the arrangement inside the autoclave also needs some precautionary measures. Dry heat sterilization. What are these? This includes your incineration. It kills by oxidation. Flaming or inoculating loop is an example of a dry heat sterilization. Hot air sterilization using the oven. This is one of the last activity in microbiology of last year. They were already in the process of sterilizing their glasswares using both autoclave and oven. Temperature required is 170 degrees centigrade for 2 hours. Oven sterilization also requires that the items be heat stable. And then we have also pasteurization. The principle, as I have made mention to you, is just like autoclaving. But pasteurization as a process is a more focus on the food that we take in, like the milk and other dairy products. Um, Pasteurization requires low temperature or low temperature pasteurization rather. Make use of 72 or below 100 degrees Celsius for 15 seconds. It kills pathogen but do not make the milk sterile. That is why you have what we call pasteurized milk. because there are some microorganisms present in milk that are helpful for the human life. The Yakult or the probiotics that you are buying, they are not sterile, but they were subjected to pasteurization, enough treatment to kill unnecessary bacteria, virus, and the other possible protozoans, but they are not sterile. I repeat, probiotics are not sterile, but the microorganisms that were left are usually chosen. Like, for example, in the probiotics, of course, the lactobacilli were allowed to be left in that food that we drink. Ultra high temperature pasteurization make use of 74 to 140 degrees centigrade, but for 5 seconds. Only as compared to low temperature pasteurization, it makes use of lower than 74 degrees centigrade, that is 72, or uh, the time used is only 15 seconds. Freezing, bacteriostatic effect only. It makes the microorganism sleep, or it does not allow any growth in microorganism, but not necessarily kill the microorganisms. It slows down the growth of microorganism. Some bacteria may be killed on the freeze-thaw process, but in the thawing process, when you are making the ice to become liquid, microorganisms could again be revived, or microorganisms could grow. Sterility is absent in freezing. Desiccation. It is a drying out or removal of water. It is only barriostatic. Once water is added, microorganism will grow. And then you have osmotic pressure, high salt. The pressure from high salt or sugar content can be preserved, or can preserve foods by preventing bacterial growth. Again, it is only bacteriostatic. These are used in most food preservation, like adding salt, adding more sugar. Molds and yeast often still grow. Then you have radiation. Ionizing radiation like X-ray, gamma particles penetrates and damages the DNA. Take note, the target of radiation is not only on the surface of the cell, but it goes towards the DNA. The target is the DNA. There are types of radiation: ionizing radiation, gamma ray radiation, short wave radiation. Continuous exposure to ionizing radiation does not achieve uh, sterility, but these are used in some food laboratories. Sterilizing plastics and other medical supplies. Non-ionizing radiation, longer wavelength, UV radiation. UV radiation is also used in some hotels in order to sterilize the rooms. Uh, of course, the main focus is the DNA. There are also some germicidal lamps that are now used. The germicidal lamps, the bulb used are usually the UV lights. They look like the fluorescent light, but what they emit is UV, or ultraviolet radiation. So this is how uh, the ionizing radiation works. You have the radiation source. This is the barriers. The barriers serve as preventing some exposure, unnecessary exposure, and it directs to the cell itself. So the focus is on the DNA molecule. Okay, this is how water swimming pools were also sterilized. Okay, this is a simple illustration of filtration in water treatment, and sometimes filtration is also used in ordinary laboratory or clinical laboratory practices. This is the ultrafiltration used in water. And we have the chemical agents. The target of chemical agents are the cell membrane, the key protein, and the nucleic acid. And these are the things that are usually seen in the laboratory and also likewise at home. In which you might not know, but these are chemicals, and these are chemicals that could kill microorganism by protein denaturation. So you have the Clorox. That is the reason uh, why during the COVID period they ask you to have a dilute Clorox solution to prepare, and then use this to wash the hallways. In fact, there are some local government units that sprayed their street with Clorox. That is to kill the microorganisms. And then, of course, you are familiar with the betadine. And then you have also the Phenolics. This includes your phenol, the Lysol, the triclosan in your toothpaste, also likewise the pisohex and benzalkonium chloride. Now, for the alcohols, you have ethanol, preferably to 70%, disopropanol, and then usually they are only used this as surface for surface purposes only. And then, of course, you have the hydrogen peroxide or the agoxinada. They attack the DNA and protein. So do not be afraid if bubbles are produced. There are some children that thinks that the bubbles produced are the microorganisms already present that had caused the infection of their diseases. No, that is not the action. Why hydrogen peroxide form the bubbles is the reaction of the hydrogen peroxide and the blood produces free oxygen. So it forms now the bubbles. We have also heavy metals, but these are now discouraged, especially the use of mertiolate or mercurochrome. That is the reason why in beauty parlors you could no longer see manicuristas applying mertiolate into the nails after manicure service. Silvers are also used by some dentists, but these are also discouraged. And then you have also the aldehydes. The formaldehydes are usually used in sterilization, um, and sometimes the formalin are not only used for good side of preservation or microbial control, but they are using this already to lengthen the life of some vegetable. There was a time that there were traces of formalin that were seen in some vegetables that are imported, or even some fruits that are imported, but it is now banned. And then you have also the ethylene oxides, and then they are used to sterilize and disinfect plastics and prepackaged devices. That is the practice now of some countries importing to other, exporting to other countries their products. So they are required to apply or expose to gases and aerosols. But usually these are halide containing. So that is my message or the last slide. Okay, thank you for being with us again in this meeting.